Right. What a joy to be here. What a joy to see all of you. This is my first time here. I'm incredibly excited, and here I'm like furiously trying to like bang together slides because I realized this group will be maybe the first group where I can actually talk about some of the math, that I get excited about, that I've never talked about before. I think it's quite accessible, and so, you know, if it goes down in flames, we will have learned something, but I promise you, I think, like, I can do it, so it must be easy math. And I think it really tells us something about what's happening with biomolecular condensates. Okay, so we're going to talk about mRNP, condensation, just a calibration here. If I say the word mRNP, how many people know immediately what I'm talking about? Okay, so many people do not, right? How about mRNA? This isn't going good. So mRNP is just messenger ribonucleoprotein particles. So the idea that mRNAs are not kind of solo in the cell, they're always covered with proteins of some sort. So this sort of stuff will come up. If you have stuff where you're like, actually, I have no idea what that is, please like fling a tomato or something, whatever's left over from the buffet. All right, so the first thing is I was trained as a theorist. My PhD is in computation and neural systems, and then I had to like kind of eat my brain entirely and go run a biochemistry lab. All the work is done by very talented biochemists. We never published an experiment that I personally have done, so it's just me with the feet on the desk, right? Basically going, yeah, do that buffer. Oh no, that buffer. These guys, particularly Caitlin, Jared, Hendrik. Jared is now in his own faculty position at Texas A&M. Put together the story that I'm going to tell you about today. It's really just a pleasure to work with these folks. This has been about a 10-year project. Actually, it was started by Edward Wallace, who's now tenured at the University of Edinburgh. So the preprint is out. I think there's a QR code at the end so that you can see this preprint, which has been out for about a year. Okay, it's the first talk. And so rather than just completely diving in, I would at least do a nod toward what on earth is it that I think that we're talking about today. This is amazing to me because Simon and I in particular have known each other for quite some time. We met in 2012, okay? And we're both like, oh, look, we're seeing these proteins do these weird things. And so it is just completely bizarre now to be old men together and to have like condensation on the cover of Scientific American, and like say what you want about Scientific American, it still is like a reasonable concern. And Philip Ball, the author of this thing, did a really nice job with this article. If you haven't seen it, it's a pretty serious bit of work. And here it is, Simon saying, these ubiquitous specs have completely taken over cell biology, and me at the end saying, this is the revolution we've been waiting for. I really believe this is true. Like absolute, like I work in the cell stress and heat shock field, and this is just the idea that melts all others. It basically provides us a way to think about what's happening in cells that utterly changes conceptually even what we think cells are trying to do, from like thermal denaturation of proteins in some toxic catastrophe that's unfolding in cells, literally, to cells are just using phase transitions to detect temperature and reorganize themselves in response to a stress that they experience countless times, right? And really countless times over their billion-year history. Okay. So to actually talk about something now, there's this phenomenon called stress granule formation, which we understand as leading to the condensation of messenger RNAs and proteins in cells when they're stressed. And may I just ask, is there like a bottle of water? Because I'll quickly, or just a cup of water someplace. Anybody have anything to even? Thank you so much. I still get nervous all this time, right? This happens in all eukaryotic life. Everywhere we've looked, if you look in the cytoplasm of cells and you have RNA binding proteins and total RNA, which is typically marked by what's called polyadenylation, right? Poly-A RNA. So this is in eukaryotes, messenger RNAs get produced, and then they get a bunch of A's, adenosines, stacked on their ends, and that's how you recognize that it's a messenger RNA. Okay, so all those things sort of clump up if you expose cells to a wide range of stress conditions. I'm going to talk about temperature today, but this is absolutely not limited, even in our hands, to temperature. Okay, so oxidative stress, withdrawal of nutrients, all sorts of other sudden changes in the environment that are generally maladaptive. Thank you, really appreciate it. generally maladaptive will lead to this phenomenon. And all I'm showing you here is, yes, that you can look in mammalian cells and you see at normal temperatures, there's basically like diffuse localization of, in this case, mRNP, which is an RNA binding protein, poly-A RNA, which is the RNA species, poly-A binding protein. They're just diffused throughout the cell, and then if you heat shock cells, this is super severe, right? Like you're not in the hospital, you're like dead at this sort of temperature, and nonetheless people use it. And you get these granules of stuff, which turn out to be co-localized poly-A RNA and poly-A binding protein and FMRP. And this happens in fly cells, not just in response to temperature, but also in response to oxidative stress induced by a chemical called arsenite. And it happens in Saccharomyces cerevisiae, my favorite organism in the universe, maybe even Simon's favorite organism. It's like we just bemoan the times we have to work in mammalian cells because yeast are so amazing. So budding yeast grows happily at 30 degrees Celsius. It gets super stressed out at 42 degrees Celsius in terms of like it upregulates a huge transcriptional response and all sorts of things happen inside the cell at 42, but you can't really see anything if you look at poly-A binding protein and poly-A RNA, right? They're still just diffused throughout the cell. But if you push the temperature a little bit higher, then you get these clumps of co-localized poly-A binding protein, so proteins, and poly-A RNA. These are called stress granules if you can see them microscopically. How many people have heard of stress granules? Okay, most people, that's a very good sign for me. Okay, that's good. Okay, so these things have been studied for decades. We think, I'm going to try to do this the right way. There is a great deal of certainty in the literature about how these things form. I'm going to try to talk you out of it, right? But what's the mechanism here? Where does the condensation actually come in? So the idea is that in response to all these stresses, one of the things that the cell does is shut down translation, particularly translation initiation, okay? And that when you do that, you can ignore this thing. This is me slapping together things, but this is all you basically need to know is that there's a bunch of messenger RNAs pre-stress that are initiating normally, and they have a bunch of ribosomes on them, okay? And maybe there's some long RNAs and some short RNAs. Then stress occurs, initiation stops, so the ribosomes stop starting, right, on the messenger RNAs. They fall off, they run off the end of the RNAs, and suddenly you have this long naked RNA. And so that long naked RNA is massively multivalent, right? And so it serves as this template or base for the formation of all sorts of RNA-RNA interactions, RNA-protein interactions. Honestly, we don't really know all of the things that are going on here. These have been intensely studied and constituted in vitro. But the general idea is that the sequence of the messenger RNA is the thing that mediates this. And as a consequence, long things offer more sites for interaction, and short things maybe don't offer as many sites, and so would be excluded from this condensation. Okay? So just to show you, because I will come back to this at the end, like how do we even look at translation initiation? There's something called a polysome profile. Now, again, just hopefully this polling is useful to other speakers. How many people know what a polysome is? Okay, and how many people have seen a polysome profile? Okay, now we're talking about half. Okay, so polysome, multiple ribosomes on a messenger RNA. Turns out that if you just look at the absorbance of RNA, ribosomes are so big and there's so many of them on these things that you can actually just separate things by size and look for bumps in your little absorbance profile, and you'll see the 40S is half of the eukaryotic ribosome, the other half, the full ribosome together, and then one ribosome, two ribosomes on a messenger RNA, 3 ribosomes on a messenger RNA, 4, 5, 6, 7, 8, 9, 10, 11. Okay, that's a polysome profile. Now, it's a sign that translation is very active if you can see a lot of these polysomes. And hopefully you can appreciate that at 30 degrees Celsius, this is data from Bernd Buchau's group, 30 degrees Celsius, most of the messenger RNA is in polysomes. Most of the ribosomes are over here in polysomes. And then as you increase the temperature, it's hard to tell that much is changing until you start to see this purple kind of deviate here, and then at 46 degrees Celsius, everything flatlines. Okay, that's the disruption of translation initiation. No more ribosomes on the messenger RNAs. Okay, so we actually know this happens in cells. What's more, the idea is that it's ribosome-free RNA. And so you can use a drug called cyclohexamide, which freezes ribosomes onto messenger RNAs, even though they're translationally inactive. Okay? Basically blocks them from being translated, but it does so in a way that leaves the ribosomes on. And when you do that, you can dissolve, sorry, this thing doesn't work because I have not plugged it in. You can dissolve stress granules. So this is minus cyclohexamide. These are these puncta, right? The stress granules at 46 degrees Celsius. You add cyclohexamide, and those things go away. Okay, so something about the ribosomes being on there seems to disrupt this process. If you do high throughput studies of what exactly is going on, then, and Roy Parker's group has done this primarily, then you find that long messenger RNAs go to stress granules. Okay, so this is their data. This is mRNA length in nucleotides. It's a log scale, so remember these things are like much, much longer over here. And these are their calls from this 2017 paper, which really spawned a whole revolution in terms of how people look at these phenomena. Where the short things seem to be less included, right? They're excluded from stress granules, and the long things seem to be enriched in stress granules. And this has been done in vitro as well, where if you take purified mRNA and protein, you incubate them with long RNA, you can get phase separation of the mRNA and protein, and the shorter you make the messenger RNA, the more those things go away. Okay? All of this hopefully paints for you a very, very consistent picture, which I'm going to try to talk you out of. Yeah? Where we got interested in this is that there are a few sort of technical reasons. The first is that I really love yeast. And this is really weird. If you think about what a yeast cell wants to do when it experiences some stress that it must have experienced countless times over its evolutionary history, which is just like take the long things and shove them into some assembly. It's like there's a whole stress response going on, right? And there's new messenger RNAs being produced, and some of them are long. Why would you just immediately take those things and shove them in some way that they can never produce a protein product? You look closer at the papers, and you're like, this has not been shown in vivo. And this didn't have a non-stress control. So what do we actually know about this? Everything I told you before, so like all the data are true, but maybe the interpretation could use some work. So we come at things a different direction than some folks. Stress granules are an intrinsically microscopic phenomenon. Literally, there's no definition of a stress granule that doesn't involve seeing it in a microscope, which is really weird, right? I mean, no associated activity. If you like ask, can you purify them? The answer is not like no. The answer is, how would you know, right? How do you know when you're done purifying the stress granule? Because there's no associated, like as a biochemist, as a pretend biochemist, sitting here, like you just follow the activity of the protein, right? Like the enzymatic activity of the protein, whatever fraction you're trying to purify out, but there's no such thing that you can do for stress granules that allows you to do that. So it's just kind of like, grab the thing and hope. I don't know. We take this approach where we're like, I don't really care what the stress granule associated messenger RNA is, but I do care what kind of condensation is happening in cells. And so you can look using other approaches that are not microscopic, and in particular don't suffer from a diffraction limit below which you cannot see things. And that is just like, well, pop cells open and spin stuff down and do that pre-stress and post-stress and see stuff that goes from the supernatant of a two-phase lysis to the pellet in a stress-dependent fashion. That just means it's like forming some sort of big particle. And there's all sorts of reasons things can form big particles. They could be a condensate, they could be bound to some other big thing, right? Like a condensate or something else. But at least you know something's happening during stress, and then you can go back and ask what's happening in the cytoplasm, what's happening in the nucleus, and so on. In particular, I'm interested in this case, where at 42 degrees Celsius, it doesn't look like anything is going on, but actually there's a ton of stuff that we know is going on. In protein condensation, we published a paper now 10 years ago on condensation of proteins at basically this temperature in response to heat shock. So here are the data that we get out, both from the previous study and our most recent study. So here I'm just looking at the proportion and the supernatant. That's, we call it pea soup, it's cute. Just the proportion of molecules that are found in the supernatant versus the pellet. Okay? And here are all the proteins we could detect. This is about a couple thousand proteins. Every dot is a protein. And you look at these distributions, 30 degrees Celsius, 42 degrees Celsius, 46 degrees Celsius, and if you just look at the distributions, it's very difficult to tell that anything's going on. I mean, the folks who are super into this, and there are a few of you out there like, I can see it's got fatter tail at the bottom. That's true. That is true. But it's like 15% of proteins that are undergoing this sort of change from supernatant to pellet. One of them is poly-A binding protein, the core marker of stress granules. Okay, so it's nice to see the poly-A binding protein, while nothing seems to be happening, it's actually doing this whole bit of business, right? Condensing. We've gone on to show that that happens in vitro. Poly-A binding protein autonomously senses temperature. It does it in RNA-independent fashion. There's all sorts of things that we know about what poly-A binding protein is doing under these conditions. We know almost nothing about the RNAs. Okay. Also, you can see that the median here is changing, right? But you really have to like squint to see it. Versus if you look at the messenger RNAs, where we just sequence the messenger RNA that we get, supernatant pellet, right, and then do the proportion of the supernatant. It's a totally different story. The entire transcriptome is like right into the pellet. Okay, first of all, that's totally different than what's previously been reported. Right? Because it's supposed to be the long messenger RNAs. And there's a bunch of messenger RNAs for which nothing is supposed to happen. Okay? So at the very least, we have a little bit of like a, okay, that doesn't make sense. Let's look at length. So take this whole bit and split it out. That's a function of length. And that's what you get. Okay, length of nucleotides, proportion in the supernatant. These distributions now split out, and I hope, and this is just the rolling average of these things. Okay, hopefully you see several things immediately. The first is stress causes the formation of sedimentable particles. I'm just going to call this condensation. If you don't think it's condensation, we can have a long conversation about all the things that we've ruled out, right? The challenge here is you can't see them in the normal way. And we would love to do things like fluorescence correlation, spectroscopy, and so on messenger RNAs, but to like our knowledge, it hasn't been done at a scale that we can use. If you know how to solve this problem, please talk to me, right? We're dying. Some people just won't believe things until they can see them, and we'd like to reach those people, yeah? Okay. So stress causes condensation. The second thing is, and it's like sort of dose dependent, right? So the higher you push the temperature, the more condensation you get. The second thing is that long things seem to go into the pellet more, right? And the third thing, hopefully, as you notice, is that happens at all temperatures, including in unstressed cells, okay? And by the way, everybody's protocol involves some sort of centrifugation if they're doing high throughput studies. Everybody's. So far in the literature, right? And so if you don't have a non-stress control, you might start to get confused about what you're seeing. So what is happening here? Yay, let's do some math. So this is physics that even I can do. I was a mechanical engineer undergrad. My PhD was in computational neural systems, but like it sounds great. But I quickly like fled away from anything related to the brain and did molecular evolution stuff. Okay, so I'm like the baseline physicist tapping on the door of physics, kind of going, will you please let me in? Okay. So let's think about sedimentation. You have a particle, it's in a tube, it's at some distance h from the bottom of the tube. You spin for some s seconds and so on. And the terminal velocity, which is the things you care about, right, for sedimentation studies, is just a function of these parameters: the mass, the rotational speed, rotational frequency of the the angular frequency, thank you, of the rotor, the radius of the rotor, the hydration radius of the particle, the viscosity, and something called, I forget what pi is. You're supposed to giggle. Okay, so two of these things are just a function of length, right? The mass and the hydration radius are just a function of length. And everything else is just some constant in your experiment, okay? And mass is like proportional to length, and the hydration radius is, it now depends on which particular papers you read, but there's some exponent on the length. Something like to the 1/3 power or something like this, okay? We're not gonna get to the level where that distinction actually matters to us. All we know is if you get all these numbers, then you can get the terminal velocity. And if you get the terminal velocity, now you can say, well, I've got a distribution of particles, presumably uniform, from the top to the bottom of the tube. And now I'm just gonna ask, how long, if I spin at this, for this amount of time, how long until this distance that they travel gets to be at least the height at which the particle is displaced from the bottom of the tube, okay? That's proportion in the supername, is like basically 1 minus this number. Okay, if you go past the bottom of the tube, you get 0. Otherwise, you get some other number. All good, okay. So how can we simplify this? Like I said, well, there's just a bunch of stuff that's proportional to length. There's a bunch of stuff that's constant. And you can just like bang a bunch of constants together, lose all of the physical intuition, because who cares, right? Because there's just two parameters that tell you what this model is supposed to be, which is something that's multiplied by L and then some exponent. Okay? And it turns out the exponent, you might think, oh, it has to be 1 or it has to be, some 1 minus the hydration radius. It turns out that even the geometry of the tube will alter that exponent. And so we just treat this as an empirical parameter. Yeah. And if you take that model, this model, and you fit it to the unstressed data, then you get something that's pretty good. Not so bad, right? Like these gray bars are so that you are kind to me. This is the top 1% of proteins by length. This is the bottom 1% of proteins by length. We get deviations in those regimes, but it's basically like no biology happening there. Okay? And inside the place where we really care about, where most of the action is, we do okay. So this seems to be, like, at least to me, to be a very satisfying explanation. You don't need to go farther than this to describe why it is that particles like mRNPs, right, messenger RNA protein particles, right, of unknown mass because they're covered with all sorts of different sorts of proteins and nobody actually knows what those things are, and there's a bunch of experiments to try to get at them, and literally nobody can tell you the weight of a specific mRNP. You don't need to know. You just need to know it's like something proportional to the length, and we get some exponents, we get these two parameters, we fit those for the non-stress case. Okay. Seems like okay. Now what do we do with this stuff? It seems curvy and I don't know. Well, so let's just think about condensation, and particularly about interactions that drive condensation. Now, I'll posit to you that there are basically two kinds that you might care about. The first is anything mediated by the sequence. Okay? RNA-RNA interactions, RNA-protein interactions, anything that just, like, by chance, some interaction site occurs along the length of the sequence. That's naturally going to be more interaction sites for long RNAs, fewer interaction sites for short RNAs. This is basically what, up until this point, everybody has been talking about. There's another kind of interaction that you might care about, and that is something that's independent of length. It's like, well, how can you have something that's completely independent of length? Well, it's still an RNA interaction, but it interacts with the ends of the RNA. Every RNA, no matter how long it is, has two ends, right? So it's length independent. We like the 5' end. I'm showing those interactions here. We technically cannot rule out 3' interactions, but hopefully if you think enough about translational regulation and so on, like almost everything happens at the 5' end. And so I would like you to walk away with that as like our best working model for what is happening. It's really the 5' end of the molecule that seems to be doing like the site of a lot of the stuff that we might care about. Okay, and this is naturally independent of length. So how do we turn this into a model? Well, I don't know. Let's just like say this Poisson interactions, right? And there's some rate of the interactions per molecule. We'll call it mu. And there's some rate per nucleotide of interactions times length. Okay, and they're both Poisson. Now we're looking at the proportion in the supernatant. And so we're going to make one final assumption, which is just if you get into if you have at least one interaction, then you end up in a condensate, and that condensate is so large that you're just going to go to the bottom of the tube. Okay? So now we're just like, 5 minutes look great. Now we're just going to ask the question, like, what's the probability that you'll end up in the supernatant, which is the probability that you'll have no interactions, right? No length-dependent interactions and no molecule-dependent interactions. And then you just get this. A nice little theory. We take the thing that we had before and we just multiply it by these two things, right? And that's the probability that you're going to remain free. Okay. 2 parameters for the no stress condition. We fit those on data before stress. And then we just have 2 parameters for the entire transcriptome, right, for each next condition. And here's what those fits look like. I don't know. Not bad, right? Like, I carry away from this. This is not like some demonstration that this is what's happening, but a demonstration that, like, the simplest thing that you can think of explains the average behavior of the data, like, to my satisfaction. And I would love to know, like, the sorts of things that we can tinker with. Of course, like, you know, those of you gazing at the residuals, and I can see that there's some structure. Yes, there is. We have no idea what that is, or there might be something interesting going on, or there might not be. But this certainly captures, like, the bulk of what we think happens. So then we can ask the question, well, how important is each kind of interaction in driving condensation here? Well, one way to tackle this is just to say, well, there's a point at which the interaction probabilities or rates become equal, right? Or we can say they're making the same contribution. And that's just the condition where L nu is equal to mu, right? That's the state where both of these things are the same. Which means that there's an effective length that you can have at which the interactions become, you know, kind of equal. And below that length, it's going to be dominated by stuff that's independent of the length. That should be obvious, right? So that means there's this equivalent length where you just divide this thing out, okay? And we can just look at this ratio and just ask, what's the equivalent length that we would expect for each of these? So, and I can, you know, the numbers are actually in the preprint. But at 42 degrees Celsius, the fraction of messenger RNAs that have L larger than this equivalent length is 17%. And at 46 degrees Celsius, it's less than 1%. Right? So this stuff is just dominated by length-independent interactions. And in fact, you can see that because in the limit of infinitely short messenger RNAs, there should be no interactions, right? No length-dependent interactions. But no matter how short you are, you always have 2 ends, right? So you'll still be able to participate in end-mediated interactions. So look what happens at shorter and shorter messenger RNAs. You still get huge differences in response to stress. And it doesn't seem to be like fanning out from some top level, right? Instead, there's just like, however short you are, you're still going to participate in these interactions. And this was, in fact, our first sign that this was the sort of interaction that we need to be looking for. Okay, if you just do a model that only includes length-dependent interactions, you fail miserably. It does, in fact, have this fan-out behavior that you would expect, right? It's just like wildly deviates from expectation. Okay, so I'll just finish up by saying, so what's the biology here? What does this tell us? The first is that, well, back to this. This actually happens. This actually happens. This actually happens. Okay? But this is stress granule formation, not RNA condensation. And it turns out that stress granules are actively, like, there's a stay away from me signal that ribosomes seem to have, which is super interesting, but it's biology, not physics, right? Where if there's a ribosome on messenger RNAs, it won't go into stress granules, but it'll still condense just fine, it turns out, okay? Cyclohexamide has almost no effect. I guess I'm not going to show you this on our on condensation of the messenger RNAs in the way that I've been showing you. we were able to take individual messenger RNAs, put little structured hairpins in the front of them to block translation initiation, and we can send them into these sedimentable assemblies as a function of how blocked they are, right, and the amount of protein that they produce. We have things where like our little hairpins produce like no protein at all versus a weak hairpin that produces plenty of protein. And these are the differences between them, like huge amounts of sedimentation that we can drive in a single messenger RNA in completely unstressed cells. We put these on a polysome gradient. And one of the things that you can do is dissolve all the ribosomes away with EDTA. This just like grabs magnesium. Ribosomes really like their magnesium, okay, so they'll just collapse away. You take individual messages, a strong hairpin. Ooh, do I even have the data? I don't. Let me elide this bit, because this is actually the bit of me sitting in the chair hoping I could show you this. But I will just tell you that we can make little messages that sit in condensates exactly in the polysome region despite being completely silent. Okay? They're in condensates that are so large that they actually appear to be ribosome bound, and yet they are completely devoid of ribosomes, produce no protein at all. They're lurking in people's polysome gradients. They've never seen them before. Okay? We can show they're actually everywhere. So how does this change our world? Well, the old world was, before stress, you have a bunch of messenger RNAs, and then stress happens, some small proportion of messenger RNAs go to granules. They do it in a length-dependent fashion that has nothing to do with biology and everything to do with biophysics, right? There's no stress specificity, and this is mostly just aggregation of messages. That really is the model. But there's an alternative. At least in our hands, this is not at all what happens. Instead, before stress, cells are happily doing their thing. Stress occurs. They're like, whoa. They take all previous messenger RNAs, and they shove them into non-translating assemblies, which are titratably pushed in as a function of how intense the stress is. They exclude exactly one population of messenger RNAs, which is the newly expressed messenger RNAs. They come out protected from condensation. They're excluded and then can be translated on their own. There's no real length dependence at all to these sorts of interactions, but it still is a result of translation initiation, and it doesn't involve stress granule formation. Okay. And I think I will stop there. There's a QR code for the manuscript splatted in a really awkward space at the bottom of this last slide, and I'll take questions. Thank you.
So I'll come around with the mic. If you're near the edges, you can just go to the mics on the edges.
Yeah, that was great. Can you go to the slide with the UTRs?
Yeah.
You went a little fast there.
Yeah.
So your point being here, whatever you put there, it doesn't make a difference. So what is the idea with this experiment?
Okay. So the central idea is like, forget about stress. Can we just block translation initiation, which we know happens in cells, and do it in a message-specific way, and then look at the fate of those messages? Okay, so we make a completely synthetic construct that we express under non-stress conditions, and we just do a little trick where we put a hairpin in the front end of that messenger RNA. This is an old trick where people have already shown, we just got these things from the literature. You can block translation initiation by putting a highly structured region that the initiation complex can't unwind, okay? And once you have that hairpin, you can make it stronger or weaker, okay? So it's a titratable thing. You just like change the delta G of your hairpin to make it go from unstructured, it's like the same amount of sequence, right? But a stronger and stronger hairpin. And then track this differential sedimentation, so if it goes up, it's condensing, okay? And those constructs just, the more you block them, the more they go into condensates, okay? And completely unstressed cells. We've also done experiments where you block translation initiation globally. The whole transcriptome goes in, right, just as you would expect. The controls here are basically like, there's a lot of work suggesting that it's ribosome-free RNA that drives these transitions. So one way to test that is to put what's called an upstream open reading frame in front of these constructs. That will cause ribosomes to initialize and then terminate on this little open reading frame, but leave the rest of the message completely devoid of ribosomes. Okay?
So what you mean is not being translated makes you insoluble. That's basically.
Not being translation
yeah, okay.
initiated.
Initiated, that makes you insoluble.
Right? But if you're not translated, You actually are perfectly soluble because initiation is proceeding normally, right, on these upstream reading frames, open reading frames. Those things, because they're not blocked at initiation, don't condense. So this sort of demonstration, which we show multiple different ways, that it's really a specific block in initiation and something about the stickiness of that complex that ends up driving condensation. And we have no idea actually what the causal molecule is. We're desperately searching for that. Yeah.
Wait. What is being recorded?
Could this just be due to the fact of the presence of the 60S, which is normally excluded from the condensate? Because if you have this initiation of translation and you have the ATS form, then this could prevent by itself the targeting to...
Yeah, so the 60S never gets, it's a great question, right? The 60S never gets on in these hairpin constructs, right? So the only reason that the hairpin actually works is that you block the formation even of the initial construct. So if you form this, for those who care about eukaryotic translation initiation, if you form the scanning complex, it'll blow through these hairpins, no problem, right? But if you bury the cap basically in the hairpin, then you can efficiently block translation initiation. So the 60S is not on there. It's not that.
So next will be Shira.
I didn't understand your explanation about sedimentation.
Yeah.
Presumably, real cell in real life is not being sedimented, it's not being centrifuged.
Most of the time, yeah. Over there,
most of the time,
yeah.
Apart from Earth's gravity. But you did not consider very simple explanation of your effect, which is entropy. The longer are your RNAs, the easier they condense without any centrifuge. And that condensation will also help them being centrifuged. So why that explanation was neglected?
Yeah, I mean, basically because that's not what we observe, right? We don't observe the long RNAs going into sedimentable assemblies at all. I mean,
you do observe that their probability to be in supernatant is reduced.
Let me take this. Yes. So in a stress-dependent fashion, we don't see it, right? We certainly see under unstressed conditions, the long RNAs go into sedimentable assemblies. That's completely true. And then, of course, what we're really, as biologists, what we're interested in is the delta between these two conditions where there's not a strong length effect. But it's true that you could just leave these things there. If you don't spin these things, then they just sit at the roof of the, right? They sit unperturbed in the centrifuge tube. So it really does require the act of very, very strong gravitational force, centripetal force, right, to drive these things into these assemblies, right? That's required. And for all of our trying, and we really did try hard, we have never seen any kind of very strong RNA length-dependent interaction, entropic or otherwise. And increasingly, folks are finding that RNAs, while you might think of them as very promiscuous interactors in cells, basically keep to themselves, that they're not constantly getting tangled with each other or all sorts of other disasters that you might find. Somehow cells seem to have figured out how to keep these very, very long, very, very highly multivalent molecules from getting into a bunch of trouble on their own.
Hi, Alan. I have two questions. So why do you think you have to dial up the temperature so much? So what do we learn from this difference between the 40 to 43 when cells would be or organisms would be dead? And also, do you think other stress conditions would produce the same outputs, like whenever it's impacting initiation?
Yeah, so this is just not, it's not temperature, it's initiation. We show this happens during ethanol stress. We show it happens during azide stress. Those things basically have very little to do with one another. We can block translation initiation under normal temperatures, and it causes this whole thing to happen transcriptome-wide, right? It's quite amazing to see. So temperature is not the thing. It's just that temperature causes the translation initiation block. Now, back to the question of why is the temperature so high? This is child's play for yeast. It experiences these sorts of temperatures all the time, right? Our cells don't. 43 degrees Celsius in mammalian cells, that's a horror show, right? Like your skin probably feels that stuff all the time, right? If you burn yourself, your little skin cells are really physiologically experiencing that, but your core cells are not. In yeast case, I mean, have a whole story that we've published little bits of, that the physiological temperature that yeast cares about for inducing a lot of these responses and making cellular decisions is getting eaten by a bird. And birds are 42 degrees Celsius on the inside. So they experience this quite physiologically, just as they're making this decision like, oh, I'm going to go on a trip, right? Travel to another part of the planet and get dispersed somewhere by animals, which is what happens. That's how they get around the planet. So they can handle 42.
Next, we'll hear from Will.
All right. Wonderful talk. So I'm wondering about a slight reinterpretation of your two fitting parameters for the length and length independent. Right, so the model you've proposed, or at least the interpretation you've proposed, as I understand, is saying that you have to have some sort of attractive interaction to make something go into the condensate. But of course, the situation's more complicated than that, and it's, maybe one possibility is that by having something really big stuck to any molecule, that is sufficient to prevent incorporation due to entropic reasons. And in particular, if the condensed phase has a very small pore size, then that would be sufficient. Yep. And then the implication being you don't have to go looking for some mystery molecule that targets things to go in, but rather just simply having something big attached is sufficient to keep them out.
Yeah, so that's super interesting. I think the first thing is you're completely right that these are effective parameters, and I don't want to conclude, because the model fits does not imply that the model is true, right? Any number of things, like even just saying that the fits look fine, right? So let's just talk about those. Those parameters can come from anywhere. Okay, so that's really important. The second bit of could you just have something big, and here I'm interpreting it as, who cares about ribosomes? Couldn't you just have anything big that would keep these things from going into? Is that the spirit of the, or at least some part of the spirit of the question?
Sure. I was thinking specifically about having one ribosome at the initiation point that is sufficient to keep the whole
yes, exactly. So this
out.
Fabulous result from, I actually forget the name of the Russian guy, single author paper, basically takes a single ribosome that uses every possible eukaryotic initiation inhibitor and shows that regardless of the length of the messenger RNA, if you just have one ribosome on the RNA, it will not go into stress granules. Which, by the way, is not what we're talking about. We're talking about condensation, which turns out, again, my failure to show you the data, but it is in the preprint. This is separate from stress granule formation. These are precursors to stress granules. Right? They're the little bits that get gathered together by cellular processes to form stress granules. So far as we can tell, stress granules, at least in budding yeast, do not form by it's phase separation of a bunch of molecules that can be constituted in vitro. It's active processes that are brought together. And apparently, having a single ribosome on any one of these messages is like a don't take me signal. Now, the nature of that signal might be physical, or it might be biological. And I think there's the interesting case of to try to get some evidence of which one is it, right? People have just done, relative to the amount of work that's been done on condensation processes, because they're exciting and physical and you can reconstitute them in vitro, versus the cellular processes, which are old and boring, and who would ever work on that, right? Involve the cytoskeleton and other things which are actually condensates, but in one dimension, right? Almost no work has been done on those transport processes relative to the other processes, and I think we're pretty desperate now in the field to kind of figure out which is which. My money is actually on active transport processes that are antagonized by the existence specifically of ribosomes and not just big things, as a way to say, for the cell to kind of logically say, yeah, I probably don't want to touch this thing because it's actively translating. And as soon as initiation is blocked and the things run off, now you're safe to come into my arms.
Okay, so now we'll hear from Sam. It might be the last question, depending on how fast it is.
Try to be fast. I just want to summarize, see if I understand. What you're saying is what's in the stress granules are not what our physics intuition would be, the longest guys, right. But the shorter guys with one protein at the end. The ones with the mostly end interactions.
The stuff in the stress granules is everybody who has an end.
Everybody who has an end.
Everybody who has
so
an
that
end.
means somehow, if I want to use physical intuition,
Yeah.
the interactions of the ends are mediating this
That's right.
phase separation. So if you look within a stress granule, do you expect to see little micelles of these mRNAs with all their ends at the center?
Such a great question. I love this. So we've never seen, I would expect to see this. I mean, another thing to say about stress granules is that they're very liquid-like in mammalian cells. We have never seen liquid-like behavior in yeast cells. So far as we can tell, the individual proteins and these things themselves, they just have very little internal dynamics. And for most people, right, look, most normal biologists, this would be a sign that the material state has nothing to do with their function. Somehow that has not been the conclusion of the field, right? But I would conclude it's like basically, and if it's true that these are mostly solid-like with very little internal rearrangement,
I didn't
then
say the micelles were solid-like.
no, this is what I'm saying. So coupling your idea here, right, which I'm taking very seriously, with the other observation that there seems to be very little internal dynamics, you should see territories. You really should see territories. And I think if we can get the imaging good enough, we'll be able to resolve whether or not this actually happens. Of course, you can't rule out that there's going to be active processes that will mix these things up at some slower time scale. But yeah, I think it's a pretty strong prediction that you should be able to start seeing substructure of these things if they're truly brought together as opposed to just some giant condensate, phase separation process that happens willy-nilly.
So sorry for the people who couldn't ask your question, but let's thank Allan Drummond one more time.